fantastic to be a part of this group now and feel that the work that we're doing is really impacting people and hopefully leading to better futures for a lot. Is there a pointer at all? No?
There is not. I'm sorry about that.
That's okay.
Just answer. Thank you.
So since the genetic discovery of POLR3-related leukodystrophy some 12 years ago, we've really learned a great deal about the clinical features of the disease and the remarkable phenotypic diversity, ranging as it does from the prevalent hypermyelinating forms to the various atypical forms, each with their own unique features, and normal or near normal myelination, as was beautifully described by Nicole in her talk this morning. And as the number of cases have increased, we've seen a growing diversity of mutations in different subunits of POLR3. And at the same time, this has been going on, numerous high-resolution structures of RNA polymerase III have been solved, initially from yeast in 2015, and more recently structures of the human enzyme have been obtained by multiple groups, including this one from Christoph Muller's lab. So these structures have allowed the mapping of disease-causing mutations and provide insight into the molecular nature of the defect in the enzyme that underlies the disease. Changes in subunit folding, subunit interactions, catalytic activity of the enzyme, interactions of the enzyme with other transcription components and DNA have all been implicated as potential defects. So beyond this, our understanding of disease pathogenesis has lagged. This is largely due to the fact that prior to 2019, there weren't systems, cell-based systems or animal models available in order for us to study the disease. This has changed tremendously in the recent years, of course. Now, as many of you know, the development of mouse models of POLR3-related disease has been particularly challenging, and this is largely because mice are not people, and they turn out not to be so sensitive to the mutations that we find in patients. So the first efforts to try and develop a mouse model, work done at McGill by Genevieve and her colleagues, were not successful in producing a mouse that actually had the phenotypes of disease. As you can see, this particular French-Canadian founder mutation, there was no disease or defects in POLR3 transcription. We were working, as it turned out, and unbeknownst to them or us, working on exactly the same mutation, trying to engineer a mouse with it. But we were able to, found out fairly early in the piece that we were working on a model that was not going to have a phenotype, and put the brakes on that work and then started to retool what we were doing to think about other ways of developing a mouse model. And this basically involved switching systems to try and move forward quickly with the research, and based on the idea that we needed more severe mutations in order to see phenotypes in mice. And so using yeast, we focused on a particular hotspot in the enzyme called the secondary channel. This is where nucleotides gain access to the active side of the enzyme, which you can barely see in red in the blow-up of the structure here. But the yellow is where all the hotspots are, and this includes the actual French-Canadian mutation as well. And so we started to introduce mutations, disease-causing mutations, into the yeast enzyme and ask what effect those mutations had on function. And just like the mouse, we found that all of the single-point mutations that we were introducing had no effect on the function of the enzyme. So we started making multiple mutations in the enzyme, and that's when we started to find effects. Like, as you can see, the combination of the singles into doubles shows growth defects on these yeast plates at various temperatures, 30 degrees, the normal permissive temperature, and higher and lower temperatures, seeing more severe phenotypes. So we settled on the mutation shown here in the red box as having sort of this intermediate growth defect in yeast, and looked at it in more detail. Inside yeast cells, we could see that there were defects in POLR3 transcription. And when we purified the enzyme from those cells and did biochemical assays, we were able to measure its activity and found it was defective and only had about 30 to 40% of the wild-type enzyme activity. So this is the mutation that we then thought, okay, we're going to put this one back into mice and see whether we can get phenotypes in the animal. And we did that, and we did our job a little too well, because when we produced these mice, the phenotype we ended up with was embryonic lethality. They didn't survive embryogenesis. So this was too strong a mutation. So what we had to do as a result of this was back off on the way we were expressing it, not from conception in the mice, but use conditional tools to control the timing and place where we turned on this mutation in the animals. We did this initially in the oligodendrocyte lineage, the 20% or so of the cells in the brain that, as Nicole described, are responsible for laying down the myelin sheath that coats the axons. The particular way we control the expression of the enzyme that's going to induce the mutation was to use a driver that was specific for those oligodendrocytes that turns on in late embryogenesis around day 11.5. These animals were viable and they had phenotypes. Now, all of this work is published, so I'm not going to go through it, but rather just summarize some of the things that we've learned. We found in these animals growth and developmental deficits in newborns, deficits in cognitive function, sensory responses, and fine motor control in adults, hypomyelination seen by various metrics, including shown down here, myelin basic protein immunofluorescence in various regions of the brain, and MRI methods as well we've used in mice to see the same thing. By transmission electron microscopy, we've seen an increase in the number of unmyelinated axons, like indicative of the loss of myelin in these mice, as well as a reduction in thickness of the myelin sheaths, which you can perhaps appreciate by looking at the bottom panels in this figure. And then finally, a reduction in the number of the mature myelinating cells that are responsible for laying down that myelin sheath. So we documented all of these properties. So the main take-homes from this work is shown here, that the disease pathogenesis involves defects that reduce both the number of mature myelinating oligodendrocytes and the ability of these cells to produce a myelin sheath of normal thickness. Now, the work on this model is continuing, but at the same time, we wanted to produce additional mouse models in order to study the disease in somewhat different ways. Remember, I said that in this particular model, only 20% of the cells in the brain actually carry the mutation, and that's essentially the oligodendrocyte population. We wanted to make a mouse model that, like the patients, have the mutation everywhere, all cells in their body. And so we had to use a different way of engineering this. We did this using an enzyme, the Cre recombinase, that's responsible for modifying the locus of the POLR3A mutation, controlling its expression in such a way with a small molecule that in adult animals, or actually not adult yet, animals that are 28 days, so they're weaned pups, we can treat them with a drug to promote a recombination event that introduces the mutation, pop out the wild-type exons here and allow the expression of the mutation. So the regimen that we've used for this is to treat with this drug that promotes this recombination at 28 days, 1 week after the animals are weaned, and then follow their growth by measuring their weight, looking at their behavior, harvesting tissue for a variety of analyses. And what we found fairly early on is that these mice exhibit a failure to thrive phenotype. They have a small size, just like the OLEG2 mice that we generated originally, but they lose body weight and they have reduced survival. So these are stronger phenotypes than we'd seen before. So we've done histological examinations of these animals, and we see reduced myelin staining in the cerebra at fairly early times, 2 weeks after inducing recombination as well as at much later times. But we've also been able to look more broadly and fairly crudely at the histology of other cells in the mice. And for the most part of the 20 or so non-CNS tissues that we've looked at, there's been nothing to see. But there was a very striking phenotype that we came across that is quite novel, we think, and that is severe exocrine pancreatic atrophy. And I think you can see, just comparing the images at the top there, at P42 and at P90, the difference in the H&E staining pattern. So the cells that are atrophying here are acinar cells, whose essential and sole function largely in the body is to produce the digestive enzymes that they then secrete into the small intestine to promote the digestion of carbohydrates, proteins, and fats for uptake. So given this fairly strong, actually very strong phenotype in the exocrine pancreas, we wanted to know whether endocrine pancreatic functions were also affected, and obviously this involves effects on glucose homeostasis. But looking at blood glucose levels either in the fasted or the refed state, or doing an oral glucose tolerance test, which is a measure of insulin sensitivity in these animals, we found no phenotype there. So it seems that we have this very strong exocrine pancreatic phenotype. This could be a mouse-specific phenotype because there are differences in how the pancreas develops in mice versus humans, and so the effects that we're seeing here in the mice could be mouse-specific. But it's also possible that it might be indicative of digestive issues, and I was struck by the comments from Nicole this morning about Wiedemann-Rauschenstreich syndrome, that they have this tremendous loss of subcutaneous fat, which is indicative of breakdown of fat, perhaps because they're not able to digest their food properly. So something to think about for the future. Now, among some of the other things we've been able to do with these mice is behavioral studies. So we put them into this behavioral spectrometer instrument where we're able to do video tracking of their movement around the chamber. We're able to look at infrared beam breaks when they're rearing up on their hind legs, and the platform that they're on is vibrationally sensitive, so we get a readout of their movements in this way. And all of these data are integrated using an algorithm that is able to define so-called home cage-like behaviors. And just to make a long story short, we've been able to identify defects in locomotor activity, risk assessment, exploratory and grooming-like behaviors of these animals. And these are all behaviors that we did not see in the other model that affects only a subset of cells in the brain, as indicated here. Now, the real advantage of this postnatal whole body model is that we can do things in it that we wouldn't be able to do in the other model. Specifically, look at the transcripts that are generated by RNA polymerase III in the brains of these mice. And we've done this fairly extensively now. So all of this work, by the way, is yet to be published. It's been submitted for publication. So this is just an example of some of the RNA analysis that we've been doing. I won't go through the details in the figures, but just highlight on the summaries. So one of the major transcripts of RNA polymerase III is tRNAs, the molecules that carry amino acids to the ribosome for protein synthesis. And we see in these mice that the precursor tRNAs, the initial molecules that are made by the enzyme, are reduced about 2 to 4-fold in the brains of these animals. Mature tRNA levels, which are the processed forms that actually function in protein synthesis, are also reduced, although somewhat less so than their precursor counterparts. But surprisingly, the entire POL3 transcriptome, which is made up of many other RNA molecules that the enzyme makes, wasn't uniformly affected. So there's selectivity in the nature of the defects that the enzyme has. And so you can see here, perhaps focusing in the panel on the right, the high bar there, that's 5S ribosomal RNA, actual RNA component of the ribosome, goes up in these mutants, reasons we don't understand. And a number of other RNAs like the U6, U6A-TAC, and RMRP, those RNAs don't appear to be affected at all. Now, all of this data was collected from animals at about 10 weeks of age, which is 6 weeks after we've already induced expression of the mutant. But we've looked also at 2 weeks after induction of the mutation, and we see a totally different picture. The only thing we see going on there are effects on tRNA. All of the other RNAs are normal. And so it seems there's a progression of disease with time, and the earliest particular change that we see is the decreased production of precursor and mature tRNAs. That's the earliest detectable defect in enzyme function that we can see in the cerebra of these animals. So we've gone and looked at tRNA expression in greater detail using a particular methodology called mature tRNA sequencing and discovered a number of important things. First, looking at the total amount of tRNA in the brains of these mice, there's a reduced level of these mature tRNA molecules. It's reduced about 25% relative to normal. That doesn't seem like a great deal, but it makes a difference in this particular instance. A 25% reduction gives you phenotypes. Now, different tRNAs, of course, are charged with different amino acids in order to decode mRNA sequences at the level of the ribosome, and these tRNA molecules are redundantly encoded in our genomes. There are hundreds of them. And so in the center panel here, you can see some of those different tRNA species sort of arrayed out, listed with respect to the different amino acids that they're responsible for carrying to the ribosome. And you can see almost without exception, everybody's affected more or less, right? Now, because of the redundancy in this system, there are multiple decoders for the same codon in the genetic code. In fact, you can collapse the number of tRNAs to a total number of 47, which are responsible for decoding the 61 sense codons that need to be read during protein synthesis. And when we collapse the data set in the center to that group of 47, you see on the right that everybody's affected. In fact, the ones on the right are those that are significantly affected, but even the ones that don't reach statistical significance, they're all affected as well. And so this seems to be a global effect on the tRNA population, which is underlying the phenotypes in these animals. And so alterations in tRNA abundance, which at P42, which is the data shown here, precedes substantial changes in other POL3 transcripts, as well as changes in the POL2 transcriptome, the messenger RNAs that need to be decoded, as well as other processes suggesting that these changes really have a causal role in disease pathogenesis. Now, we've also looked at the POL2 transcriptome, the expression of the messenger RNAs that need to be decoded in the brains of these animals, and found a couple of striking results. And I'm just going to show a few of those here. One is the expression of genes that are specific to neurons, the messenger RNAs that are really only found in neurons that are a marker for those cells, were dramatically reduced in the brains of these animals. Now, this is not because the POLR3A mutation has a direct effect on the expression of these genes, but rather that the cells that contain these messenger RNAs have reduced abundance in the brain. And that's evident from the immunohistochemistry we've done looking at nu-N staining. This is a neuron-specific marker, and we see reduced fluorescence intensity and reduced numbers of neurons in the cortex of the mice, indicative of the loss of these cells in the animals. The other striking finding was an increase in the expression of messenger RNAs that encode proteins important for the innate immune response and for cell death pathways, shown here in the sort of middle-ish heat map. And this sort of suggested to us an upregulation of the innate immune response in these animals, which we confirmed also by immunofluorescence using this marker EBA1 for microglia, which are the resident immune cells in the brain. And you can see there's a dramatic increase in the number and morphology of those cells in these animals. And so the activation of the microglia, we think, is promoting an inflammatory response in the mice and leading to neurodegeneration, and this is how the disease is progressing. But the other thing that we also see that's worthwhile pointing out, I think, is that there are regional differences in the sensitivity of the brain to the particular mutation that these animals are carrying. And we see much stronger effects in the striatum, for example, than we do in the cortex. And so these sort of regional differences are going to be important in understanding particular phenotypes that result. Okay, so here's the summary. So as of right now, we have 2 POLR3A mutant mouse lines that we've been able to develop. The OLEG2 mice, this is a developmental model. It allows effects of the POLR3A mutation to be studied in specifically the oligodendrocyte cell population from embryonic day 11.5. And these tamoxifen-induced mice, this postnatal whole body model, where the effects of the mutation can be studied largely in a post-developmental state, but in any tissue or cell in the animal. The key finding, I think, at this point is that diminished tRNA synthesis is the primary defect in the cerebra of this whole body mice, and this is indicative that, you know, there are going to be effects on protein synthesis, and the expression of proteins perhaps is the primary initial cause of the neurodegeneration that we see in these animals. And finally, as a result of working with these mice, we've been accumulating numerous cellular and molecular readouts that can be used for preclinical testing of genetic and pharmacological approaches that are aimed to mitigate the effects of POLR3 related disease. Okay, so I have important acknowledgements to make. Excuse me. Our research team at Einstein, Robin Moyer, who is here at the meeting, Emilio and Christian, our collaborators, particularly in the Stanley Lab, and Violetta Chitu with respect to the microglia studies, our resident pathologist for this work, Bridget Shafit-Zagato, and numerous others involved in various aspects of the work, the imaging, the histopathology, animal behavior, the epigenomics, and so on, all been vital and participants in this work. And we've been at this for quite a long time. We started this work in 2016 with support from NIGMS to make the mice. And this work has been carried forward with grants from NICHD and NINDS to advance our understanding of the 2 models that I've talked about today, along with support from Einstein in the way of pilot grants from their IDDRC center. Obviously, I have to acknowledge the Yaya Foundation for their support as well. And, you know, I'm happy to say that just very recently, we've also received a brand new grant from NINDS that's going to continue to fund the work that we've been doing for another 5 years. So we're extremely grateful for that work as well. So, thank you very much for your attention.